Next talk is a team talk from the Allen Institute, and it's looking at some incredible data from electron microscopy. I'm looking at the Chandelier cells. The title is Enlightening the Chandelier, and it's presented by Agnes, Bodor, Cc, Schneider-Mizell, Nandi, and Zhuang.
Thank you. So we asked the question, what is the logic of the inhibition in the neocortex? And when and how inhibition can affect cortical circuits. To answer this question, he used a very particular cell type, the Chandelier cell, which has very distinct morphology and also connectivity. And because of these features of this cell, it's a very good candidate to examine this cell type through many pipelines in the institute. Since the connectivity is very unique, it's possible to know every synapses of this cell type, which make it possible to test the effect on pyramidal cells, the target cell of this cell type, in a model. And then, since the genetical profile also unique, we are able to label this cell and test the hypothesis regarding the effect of the Chandelier cell on pyramidal cell in a live animal. So here you can see the cell, the Chandelier cell. The population we examined, it's in mouse primary visual cortex, and it lays in a top of layer 2, actually between the border in layer 1 and 2. And you can see the dendrites go up to layer 1, and all the axons below the soma occupying layer 2, 3. And when you look detailed, you can see these vertical lines, which reminded the early discoverer to the candles of the chandelier. And when you look this structure carefully, these are small chains of synaptic terminals. And as you can see, they are always vertical, so it suggests that it might target a vertical structure in the cortex. So actually, most of the morphological cell type was discovered early, but this cell type, Chandelier cell, is an exception. It was discovered only the early 70s by Santagotay and Jones in the same time, but independently. This is an original drawing from János Santagotay, the Hungarian neuroanatomist. And they try to draw the whole, the structure of the neocortex. You can see the layers, also the pyramidal cells, and you can see the inhibitory cells within. And you can see this cell type here, labeled as CHC, which is a common abbreviation for Chandelier cell. And you can see the structure I described. You can see the soma, the axon below, and targeting on the drawing some vertical structure, which is the apical dendrite of the pyramidal cell. But if you look at the drawing more carefully, you see another cell, which look exactly as I described the Chandelier cell, and that's labeled as axonic cell. The only difference is here, the candles targets the axon initial segment. Later studies shows these two cell types are the same. And it only targets exclusively the axon initial segment of the pyramidal cells. So in this talk, Casey and I will use electron microscopy to examine the logic of the Chandelier connectivity. Then Annie will use modeling to examine the effect of, based on our EM data, on the target cell, the pyramidal cell function. And then June will use a live animal to test the hypothesis based on the modeling in live animals. I introduce here the electron microscopy dataset we have. You can see we carefully mapped the visual cortex, then sectioned the tissue to 40 nanometer electron microscopic sections. And what is really unique about this dataset, theoretically we can see anything which is visible in electron microscope, all the cell, all the axons, all the synapses. And here you can see a pyramidal cell from this data set. And also we have a tool to automatically detect every synapses. And you can see here the incoming synapses with greens on the dendrites and the soma and on the axon initial segment. And through the talk, we will use this form to, just for clarity, we only show the soma and the axon initial segment with the incoming synapses. Here I show 4 nearby pyramidal cells from our data set. And you can see the Chandelier axon targets each of these axon initial segments differently. Some didn't receive any, and some receive more, and some receive less input from this Chandelier cell axon. On average, each button mostly forms just one synapses. And you can see here another axon. It's doing the same thing. It's target another set with another different amount of synaptic terminals. And here you can see every single Chandelier axon in this data set, which target these 4 pyramidal cells. And you can see each axon initial segment receive a different amount of input in a different combination of these axons. And of course, we don't have only 4 cells, but we have almost 150 good axon initial segments of a pyramidal cell in this data set. And also, we have all the Chandelier axon target this cell. So we can see, theoretically, the entire connectivity of the Chandelier cell. And in the next case, we will talk about is there any logic, any particular system how this axon targets the given cells.
Thank you, Agnes. And thankfully, the answer is yes, there is a bit of logic. So despite this amazing specificity with what Chandelier cells innervate the axon initial segment of pyramidal neurons, not very much is known either about what function they're performing in visual processing or cortical processing in general, nor is much known about the network structure of how populations of Chandelier cells talk to populations of pyramidal neurons. And the EM data set's a really good place to start looking at this because we get just exquisite knowledge of where cells are in space in relation to each other in the same nervous system, in the same biological tissue, as well as beautiful, precise connectivity maps where we know for every one of the axon fragments that we have reconstructed in EM, we can tell you exactly how many synapses it makes onto specific pyramidal neurons. So the question is, can we put this together to understand something more about the population architecture of how Chandelier cells talk to pyramidal neurons? And the first question that we decided to ask was, if you're a pyramidal neuron, how much Chandelier inhibition do you get anyway? When you look at a histogram of this measure, you see something that looks maybe surprisingly wide to you. That there are some cells that are receiving 0, 1, maybe 2 synapses of Chandelier input. They're basically escaping any inhibition from Chandelier cells, whereas other cells are receiving maybe 2 or even up to 3 times the average amount of Chandelier input. So some cells are getting slammed by inhibition potentially, and others are escaping it. And this is extremely different from the other major source of inhibition onto these neurons, the synapses that pyramidal cells receive on their somas. Here, if we take the exact same cells and we count the synapses on the soma, you see that not only do all pyramidal neurons receive plenty of somatic inhibition, the sort of most structurally inhibited cells, as it were, are maybe getting 30% more than average. So there's something different about the amount of variability in Chandelier inhibition compared to other sources of inhibition in cortex. So we wanted to understand if there's any structural properties that could help understand what's going on here. And so for every pyramidal cell, I computationally cut out the soma and a constant part of the axon initial segment. And for these bits, we could measure a number of properties about the location of the cell, the size of the cell, the number of synapses it's receiving, and then as well the AIS, we can get information about the size of the axon initial segment, and then also inputs from some non-Chandelier types that are sort of rare but interesting. If you take a data-driven approach to the measurements of all of these properties across the 150 or so pyramidal neurons, you can actually get these sort of data-driven mixtures, sort of doing sort of something like principal components analysis that seem to be very interpretable, that there's one component that corresponds to how much inhibition the cell gets from other sources of cell types, other cell types than chandelier cells, one component that corresponds to how big the cell is, and one component that corresponds to how deep it is within layer 2-3. So for every pyramidal neuron, we can use this components analysis to basically make 3 numbers, one giving an indication of how big the cell is in size, one giving how much inhibition that cell is receiving, and one where the cell is in depth within layer 2-3. So now the question is, do these 3 structural measures have anything to do with the amount of chandelier inhibition the cell is receiving? So the simplest approach to this is just ordinary least squares, right? Is there a linear relationship between these values? So I can fit this to the data, and I can express these coefficients. I'm going to express the coefficients in this horizontal plot. So a positive coefficient here means that there's a positive correlation between this value, in this case soma inhibition, and the number of chandelier synapses that a pyramidal neuron receives. So what this means, for example, is that the more input a pyramidal cell gets from other inhibitory cell types, the more chandelier cell input the same cell receives. Size shows a weak but statistically significant positive correlation, and depth is a strong correlation that's quite negative. So the deeper you are in layer 2-3, the less chandelier input you receive. So great, there's some interesting structure here, but synapses alone are a pretty aggregate measure. Here, for example, are 2 different ways that you could receive 8 synapses if you're a pyramidal neuron. And they might be very biologically distinct. One involves a single axon that puts 8 distinct synapses on you, and the other involves 4 different axons, so 4 different connections, each putting 2 synapses. And these might be biologically meaningfully different. Because the data lets us actually trace the identity of every one of these axons independently, we can measure these 2 values separately for every axon initial segment and get both the synapses per connection and the number of connections for every pyramidal neuron. Now, if we run the same analysis, we see no significant correlation with the structural properties and the average number of synapses per connection, but we see extremely strong relationships with the number of distinct connections, suggesting that the structural heterogeneity in chandelier input is driven much more by a number of distinct axons targeting the pyramidal neurons and less by the synapses per connection. So what's interesting about this is that once you start thinking about connections, you have to start thinking about geometry. Because if I showed you these 2 pyramidal cells in gray and asked you which one is more likely to talk to this chandelier axon fragment here in red, you would be pretty quickly say, well, this one's close enough to target it, and this one's not. So this is a very different realm of connectivity than here. So to take this intuition and formalize it, we went back to this exquisite anatomy data, and for every axon initial segment could look around a given radius of, in this case, 7.5 microns, and ask how many chandelier axons are within that radius. And that will be what we're going to say is our number of potential connections. And then of those potential connections, what fraction of them actually form synapses onto the axon initial segment? And that will be the connectivity fraction. As the product of these 2 values, that's the number of connections. But we can measure them independently. So when we do the same regression analysis, we start seeing something really interesting. If you look at the number of potential connections, you no longer see any relationship with somatic inhibition, but you still see this negative correlation with depth. So that suggests as you go deeper in layer 2-3, there's just fewer chandelier axons around. In contrast, if you look at the connectivity fraction, the number of nearby cells that are actually recruited to form synapses, then you no longer see any modulation with depth, but you see this positive correlation with somatic inhibition, suggesting that the more inhibition a pyramidal cell receives at the soma, the more it recruits the nearby chandelier cells to connect to it. Now, this model that I just described implicitly assumed randomness, that a chandelier cell and a pyramidal neuron didn't actually care who was who when they formed connections. And so we wanted to test that. Is this connectivity completely intermixed, or are there some chandelier cells that prefer to talk to some pyramidal neurons and other chandelier cells that prefer to talk to other pyramidal neurons? So to measure this, we basically asked how often is this particular motif where two chandelier neurons both try to talk to two pyramidal neurons, how often is that last edge connected in the case that this is a geometrically allowed connection in the sense that I just described? And we could compare the likelihood of this connection existing with a shuffled network where we maintained the same geometric structure of proximity between chandelier axons and pyramidal cells. And what we saw is that there's actually no real difference between the observed value and the shuffled network, suggesting that there's no evidence of target selectivity beyond the geometric proximity between neurons. So we were able to put together some facts about the logic of chandelier connectivity at the structural level, but to interpret this in terms of processing and what the animal is doing, we needed to know more about function. And so for that, we turned to our colleagues who make amazing biophysical models of how neurons work. And Andy's going to talk about what he found there.
Thanks, Casey. So we wanted to understand how this chandelier projections affect the layer 2-3 pyramidal cell in mouse visual cortex. And for us to probe that question, we needed to simulate this pyramidal cell with synapses. And for that, we needed biophysically detailed models of these pyramidal cells. And before I go into modeling, I just wanted to point out that this would not have been possible because of, unless we had this in-house pipeline project, namely IVSCC, which involves morphological reconstruction, wholesale patch clamp recording, and then packaging all this amazing data into an API. And all of this data is publicly available on this website. So going back to the modeling, what I mean by single neuron modeling is distributing synapses, distributing conductances along the morphology of this neuron such that the model defined by these conductances and the morphology replicates the experimental recordings shown here. And we adopt an evolutionary algorithm to fit these conductances. Here I'm showing the algorithm in action. What it does is, in an automated framework, generates a massive number of models by sampling from the parameter space shown here, and then evaluates this model against the experimental recording until a suitable match is detected. And for a before and after shot for this modeling, here is how the model matches the experiment at the first generation, and here is at the end of the optimization. So on the top left, I'm showing a snapshot of how this model performs against the experiment. So here you can see the FI curve, the subthreshold and spiking responses, and as well as the AP shape. So now that we have the model, we are in a position to distribute synapses on this on the morphology of the neuron, especially excitatory synapses on the apical dendrite, and inhibitory synapses mimicking the chandelier projection on the axon initial segment of the pyramidal cell. And just wanted to also point out this is a collaborative work with my colleague Tom Chartrand within the modeling group. So now that we want to distribute this chandelier synapses on the axon initial segment, we don't have to make assumptions anymore. We have the EM data to inform us about how this synapses are being distributed. For example, if there are 8 synapses, I'm showing here that there are 3 different presynaptic chandelier terminals that are projecting this 8 synapses. And we call this EM-driven because this is the mostly common scenario we see in the data, that is each pyramidal cell getting inhibited from a number of different presynaptic chandelier cells. And we compared this particular configuration with 2 other configurations, namely synchronous, where this same 8 synapses coming from one particular presynaptic chandelier terminal, and asynchronous, where this 8 synapses coming from 8 different presynaptic chandelier terminals. And here I'm showing in the movie that how the presynaptic chandelier spike trains are inhibiting the pyramidal cell dynamically. So I have now laid out the 3 different configurations here, the EM-driven, synchronous, and asynchronous configuration. And also for comparison, I have this green case where there is no inhibition. So just for an example, as you can see, when there is no inhibition versus when there is inhibition, the spiking output of the pyramidal cell goes down. So when I compare this 3 different cases with both excitation and inhibition, what I see is across different level of inhibition regime, the asynchronous case, that is when the synapses are coming from different cells, is the most effective form of inhibition in inhibiting the pyramidal cell spiking. The synchronous case is the least effective form of inhibition, and the EM-driven or the biological case is mediating between these 2 cases. And so this is providing some sort of a trade-off between the most effective form of inhibition, represented by the asynchronous form, and the least effective form of inhibition, represented by the synchronous form, because the synchronous case only offers 1 connection. So next, we saw in the EM data that this chandelier projections are axo-axonic. So naturally, we wanted to understand whether there is any difference between the axonal projections of the chandeliers and the somatic projections. So what we did is repeated the same simulations, but now instead, along with the axonal inhibitory projections, we also have somatic inhibitory projections. And here I'm showing on top the excitation and inhibition, excitation in black and inhibition in red. And here in the 2 rows, I'm showing the raster plot for the pyramidal cell. On top is the axonal, and bottom is the somatic. And here you can see that when the excitation and inhibition are offset by some time, the pyramidal cell spiking output between the 2 cases on top and bottom are comparable here. But when the excitation and inhibition are coactive, that's when the axonal inhibition is much more effective in reducing the spiking of the pyramidal cell than the somatic inhibition. So if we focus on this particular case and look at the statistics of in our simulation, we see that in a moderate inhibition regime, this axonal inhibition is at least 20% more effective than the somatic inhibition. And this moderate inhibition regime is also representative of the chandelier synapse distribution that Casey showed earlier in his data. So from my simulation study, the prediction is that the chandelier projection is most effective. Chandelier axonic projections are most effective when the excitation and inhibitory barrages are coactive. But what are these chandelier cells doing in vivo? And for that, we turn to June.
Thank you, Arnie. My name is Zhuang. I'm a physiologist. So after seeing this beautiful EM reconstruction, the quantitative analysis of their connections, and the nice modeling work to predict their effectiveness of inhibition, you will understand why I'm so dying to see their activities in vivo. But because this is almost purely morphologically defined cell type, and they are extremely sparse in cortex, so historically it is very, very hard to target those cells in live animal. And thanks to the previous team talks, and I think you've been already convinced that we can target chandelier cells in vivo. But here I'm just going to show you once more, as a use case of how to use Allen Institute data product to guide your own study. So our large-scale transcriptomic study have indicated there's one gene may be related to Chandelier cell, which is called VIP receptor 2. I'll call it Viper2 gene. And if you go to our data portal and type in this gene, you will see, okay, this gene, among all these more than 100 transcriptomically defined cell types, Viper2 gene only expressed very tightly concentrated in one of the cell type, which is a subtype of parvalbumin positive cells. And are those cells really Chandelier cells? We go to another data set, IVSCC, which has been introduced by other team talks and Ani. This time we are looking at Viper2 positive cells. So here is an example of the fully morphologically reconstructed such a cell, and those are their in vitro physiology. You can see the nice chandelier morphology of their axon, and indeed faster spiking. And then for all the Viper2 positive cells, 92% of them are Chandelier cell. For all the morphologically fully reconstructed, 92 are Chandelier cell, and 32% in the primary visual cortex are Chandelier. So basically, almost all the fully reconstructed Viper2 positive cells are Chandelier cell, except for just one cell. So hopefully, I can convince you that now we can target specifically the Chandelier cells in vivo. And we made this mouse line, so it suddenly opens the door for all sorts of in vivo experiments you can do. So the most easy strategy will be you just inject Cre-dependent whatever target gene into cortex, and then you can into the Viper2 Cre mouse, and then you can label those cells and do whatever you want. So first, we just inject the GFP, a structure label, and here is in vivo 2-photon Z-stack. So you can see the dendrite, the cell body, and below the cell body, you can see those little punctas just everywhere, representing the candles. And if you do histology, you can see almost this textbook-like morphology of Chandelier cells. And same thing is true for if you inject the calcium indicator GCamp. And you can see the cell bodies lies between layer 1 and layer 2, and all the candles are in layer 2, 3, just resemble the EM morphology. So since we have the calcium indicator in the Chandelier cells, of course, we want to image their activity. So we leverage another large-scale pipeline, which you will hear tomorrow. So I'm not going to describe this in detail. So this pipeline is called the Brain Observatory, allows us to do in vivo 2-photon calcium imaging. And one thing I just want to point out is this setup allows us to record the pupil and the running speed of the mouse at the same time. And you will see later why this is important. Okay, so there's still one thing left. It is like, if you look at the cell body distribution in the cortex, you can see they are extremely sparse. If you just do a regular 2-photon imaging, a single plane, you probably will see 2 or 3 cells per imaging session, which is very low throughput. So here, thanks to the Institute, another effort to develop state-of-the-art imaging tools, in this case, Deep Scope, which uses a spatial light modulator very quickly to switch the depth of your focal plane, allows you to do almost simultaneously, multi-plane imaging. So we can, in this case, we can image 3 different planes at the same time, significantly increase our throughput. Okay, so finally, we have all of our tools together. So we put a GCamp6F into the Chandelier cells, and we use volumetric imaging with simultaneously recorded 3 planes with 16 microns apart. And we reached the volume rate of 12 hertz. And this is just, to my knowledge, will be the very first in vivo recordings of Chandelier cells. So here it goes. Okay, I don't know if you have noticed it or not. To me, everything just lights up at the same time, right? And you can not only just see the cell body, but also those candles are just like lights at almost the same time. But this is 20 times speed of the real time, so those are actually pretty slow events, okay? And the good thing of this pipeline is just after every imaging session, I can package all the data into a standard data format, namely neural data without borders, NWB file format. So and then I can, with this file format, I can just very easily share this data with other people. So in this case, I just send this data to another scientist, Brian, with very small amount of explanation. He understands the data, he can start to analyze it. So here is the calcium traces you have just seen from this movie, from those in total only 5 cells. You can see they are really highly coactive. And to quantify this, we did, Brian did the pairwise cross correlation. You can see the correlation coefficient is about 0.5, which means they are really very highly coactivated. And what about the other cell types? We use the same data analysis and compare the other inhibitory cell types, PV cell, VIP cell, and SSE cell. And the Chandelier cells represent a cell type which has the highest correlation, even if it is just a subset of the PV cell. Okay, so now you wonder, like, during this highly activated period, what does the mouse do? I'm not showing anything on the screen. It's just dark. They are just sitting in the dark. And given that their cell body is in between the layer 1 and layer 2, and the superficial layers, which receives a lot of input from the neuromodulatory axon inputs, so we hypothesize these activity may be brain state related. And thankfully, we have the recordings of the mouse pupil and the running, so we can just plot them together. These are the running and the pupil traces. So really, everything just happened at the same time. Okay? And if you zoom in to this little period, and when the mouse pupil already dilated, but it hadn't started to run yet, the chandelier activity already catches up, meaning that this is not just motor-related activity, but indeed it is. Indeed, the chandelier cell is representing activity related to the innate brain states, and the quantitative analysis showing you, indeed, the chandelier activation is more correlated to the pupil than the running. So these is consistent very well with the modeling work of ANI, meaning if the chandelier cells inhibition needs to be more effective, it needs to be happen during the period which you have a lot of excitation, which is exactly what's happening during arousal. So these conclude the in vivo part, and now to wrap up, I will hand the mic to Casey. Oops, sorry.
No, good. All right. So I'd just like to take a couple of minutes at the very end here to pull these sort of in threads together and think about how what we found about chandeliers might affect visual processing and cortical processing in general. So what June just showed was that this pupil response, which is thought to be a strong correlate of an internal arousal state, that this arousal state drives collectively these chandelier neurons, and it's, as June just said, it's known from the work of David McCormick, Andres Tolias, Jessica Cardin, and probably others, that it's exactly during this period that there's widespread depolarization on the excitatory neurons in this network. Ani showed that the inhibition from chandeliers onto the layer 2-3 pyramidal neurons is particularly strong and is really well tuned to shape their activity, except and then what we showed is that this inhibition isn't just distributed uniformly through the population, but is actually highly structured. First, forming a stronger to weaker gradient across the depth of layer 2-3, possibly creating a sort of less excitable and more excitable subpopulations within within the layer, and also respecting cell-specific inhibition amplitudes that might potentially reflect some sort of inhibitory homeostatic balance. And so taken together, these chandelier cells really seem to be a key piece in reshaping the inhibitory structure of cortical processing in layer 2-3 during these particular periods of arousal, and of course potentially other roles as well. And looking forward, we pulled function, anatomy, and modeling all together to analyze one particular cell type to cell type interaction. And we're on the cusp of getting data that's really going to let us take this integrated approach and interrogate deeply a rich menagerie of cell types and their interactions. And we're looking forward to that in the years to come here. So with that, on behalf of everyone, I'd like to thank really a small army of people working on EM, on modeling, on computation, on in vivo imaging, and all of the teams here. Of course, our founder, Paul Allen. And with that, we would love to take any questions you may have.
Thank you, team. And there's a question up the top, and a microphone is on its way.
What do you know about, I mean, two questions. A, do you know anything about gap junction? I realize the cells are quite apart, but given the correlated activity, do you know anything about gap junction among the...
They are gap junctionally connected, yes. The chandelier cell dendrites are gap junctionally connected with one another. It's known in the literature, and we also see it within even the very small EM volume we have.
So how much of the cross-correlation can be explained by the coupling?
I have no idea about that. I don't even know how you'd get into it. Does it matter if the functional responses are, if the spikes are?
Well, it's important to understand how much is given by common input.
Yeah.
You know.
Yeah, that would be great to study in the future, but we don't know that right now.
And then in the, I guess it's a question for Neil, in the modeling, do you assume that the axonal initial segment has a different distribution, has a high density of sodium channels, or they're shifted towards lower potential, lower thresholds compared to the somatic fast sodium channels?
Yeah, we made sure to constrain the spiking initiation at the axon. And the sodium density is higher at the axon. Also, the AP threshold is lower compared to soma.
Okay, maybe we can do one more question. Or
wonderful data. It's very interesting. One thing I'm curious about is whether you have looked at whether the pyramids that get inhibited by these chandeliers, do these pyramids excite these chandeliers, the very same ones? Were you able to look at that?
Some do, some don't. It's known, so the volume that we're studying is a little too small to get full axon collaterals on the pyramidal neurons. So for some of them, we absolutely do have this feedback. There's data from a Zhuang paper a couple of years ago that does show actually that there's interesting local pyramidal cell to chandelier feedback. We can't, unfortunately, our data set's a little too small to give their result, but we can show certainly the existence proof of excitatory feedback. Yeah.
okay, and we can do one more question. Yeah.
It actually sort of relates to that. So given the arousal modulation and the gap junctions, I mean, maybe this is seeming like it's sort of a more globally oriented population. But along the lines of the last question, it might be hard to look for direct connectivity because of axons moving in and out of the volume. But would it be easier to look at common input, like axons that are local, synapsing on the same connected pyramidal in chandelier cells?
That's absolutely a thing we could do, I think. So this pinky volume that was described now is kind of our phase 1 data set, and we are moving kind of quickly in the next coming months towards millimeter scale EM. And so I think some of these questions would be better suited to just waiting until the data's there to really answer the question right.
Just one quick thing to add on to Christoph's question about gap junctions. There are some blockers like mefloquine that are not perfect, but that can do some modulation of gap junctions. You could hypothetically do the physiology experiment, but inject mefloquine into the animal and look to see if the synchrony changes.
I would say not with the calcium imaging, because calcium imaging is too slow to be able to tell for sure if they are gap junction connected. And then since they are very sparse, so only thing I can think of is you do slice work, you label the chandelier cells and patch them and patch both of them to see if they are, am I right?
Okay, we can do another round of applause for the team.